Hi, everybody. So I want to take an opportunity to kind of break glycolysis down into its most basic components to give you a jump start on how to study and understand what's going on. So this is going to be a review of glycolysis. And if you want to follow along, not only can you use the lecture slides, but in your biology and focus textbook, you're going to see a review of these reactions on pages 146 to 147. And this is a way for you to follow along with the full molecules. So the way you're going to be tested is by seeing these full molecules. But remember that you're only responsible for the things that I'm going to be talking about in this video and that were highlighted for you in class. So let me give you an example. We know that glycolysis is going to begin with a molecule of glucose. And if you want to break down glucose into the things that you need to know, we can say that this is a 6-carbon sugar. So we now have our representation here of glucose as a 6-carbon sugar. Now, the first step of glycolysis is going to be when an enzyme adds onto our 6-carbon sugar a single molecule of phosphate. But we know that if we're going from a molecule that has 6 carbons of sugar to 6 carbons to 6 carbons plus a phosphate, we need to have a source for that phosphate. And in this reaction, it's going to be that ATP hydrolyzes one of its carbon or one of its phosphates and yields an ADP. So it's actually going to be this molecule of this phosphate here, which gets transferred over here. You may or may not recall from class that we named the kinds of enzymes that are responsible for transferring a phosphate as kinases. And if you were to take a look in your book, you would see that this is in fact a kinase. If you noticed an enzyme here named a kinase, that would also be a big clue to you to know you were going to transfer a phosphate. But since you don't need to know the name of this particular kinase, I'm going to leave it out of our schematic. The next step here is that we've added some energy to this glucose by adding a phosphate to it. So we're going to have some rearrangement of the carbon atoms, but it's still going to be yielding for us this same 6 carbons and a single molecule of phosphate. So in terms of your understanding of this molecule, nothing has really changed. The following step, however, is that this top carbon up here is in a new position, and that new position makes it primed for the addition of another phosphate. So we go from a 6-carbon molecule with 1 phosphate to a 6-carbon molecule with 2. And just like how in our previous steps, we had the source of this phosphate was an ATP, we have another hydrolysis of ATP, which is going to be the source of this second phosphate. The difference is that while this is in fact going to be a reaction catalyzed by a kinase, this is a name that you are responsible for knowing and can give me, let's say on an exam, which is phosphofructokinase. That name comes from the fact that this 6-carbon molecule with a single phosphate on it is actually going to be something that is known as fructose 6-phosphate. So this kinase is adding a phosphate onto a molecule with that very similar name. Now, at this point, we're actually going to use this additional bit of energy that was added into the molecule, that additional bit of disorder from that second phosphate is going to allow us to split this molecule right down the middle. We're going to basically like fruit ninja this thing in half. And when we do that, what happens is we now have 2 3-carbon molecules. 3 carbon sugars are known as trioses, and each one of them is still going to keep a single phosphate. Now, at this point in the process, we've actually gone ahead and invested some energy. I've spent an ATP here and an ATP here. So we're actually -2 ATP in terms of the energy in this reaction, which is why all of these reactions so far are known as part of the energy investment phase. What's going to happen now is that we've invested all of this energy, but we need to finally start getting some energy payoff. So what we're going to do, the very first part of this reaction of this phase is going to be the most complicated. And in order for us to really understand it, we need to take a look at things that are happening simultaneously, but we need to start understanding them as single components. So let me remind you that there are two different molecules of this 3-carbon here, which are known as G3P. This stands for glyceraldehyde 3-phosphate, but you, as one of the exceptions to this rule of don't use abbreviations, are only responsible for knowing G3P. So everything that happens from this bottom part is actually happening twice. But let's start with just our 3-carbon molecule of G3P here, and I'm just going to draw it once. So if I were to pay off energy, I could remove this phosphate using enzyme and substrate level phosphorylation to remake one of the ATPs that I had already made. But that would mean that I invested 2 ATPs into the system, and then I just get 2 back out. It's basically a net zero. And the cell doesn't want to do that. So it's going to get creative here. And it's actually going to use an inorganic phosphate, written as P with a little I. And this inorganic phosphate is actually going to be a phosphate molecule just as you know it. But it's not attached to any carbon. Since carbon is what makes things organic, this is an inorganic phosphate because it's floating around, not attached to anything else. So we're actually going to attach this phosphate here onto our G3P, making it into an organic phosphate because it's now actually attached. So what was our inorganic phosphate is now organic, which is kind of cool. And it's, as you're going to see, helps the cell get some payoff for this system. But you can't just do this without any manipulation. So what's going to happen is that we're going to need to pull some electrons from G3P, which creates the space and the availability for the phosphate to attach. You are not responsible for really getting the chemistry that's here, but you do need to know that this step occurs. And the idea is that if you need to pull some electrons here, well, we know one enzyme which is really good at that, which would be a dehydrogenase. Dehydrogenases are paired up with molecules that are called electron carrier molecules. So we're actually going to input a molecule of NAD plus here and get out a molecule of NADH, stealing these little electrons that we have over here floating around. These are going to get stolen and attached to NAD plus by this dehydrogenase. Now, just like how you have one kinase molecule that you need to know, you have one dehydrogenase molecule that you know. And this one is triose phosphate dehydrogenase. Just like how our kinase is named for the phosphorylated fructose molecule that it acts upon, here we're naming this for the 3-carbon phosphorylated sugar. So this is a triose phosphate dehydrogenase. Now, just like how when we started this reaction, there was a really nice pattern where we went from glucose, hydrolyzed an ATP, now we had a 6-carbon with a phosphate, hydrolyzed another ATP, we had a 6-carbon molecule with 2 phosphates. This part gets very, very repetitive. We now have a fully loaded, energetic 3-carbon sugar with 2 phosphates. So the very first thing that we're going to do is remove one of those phosphates. But I wanted you to pay attention to the arrow here. Now we're taking an ADP we're going to take this sugar, give it to the ADP, which makes ATP. So now, instead of having a 3-carbon sugar with 2 phosphates, I have a 3-carbon sugar with 1 phosphate. The following step, after some rearrangements, which again, for you, don't really change anything. There's still 3-carbon sugars with phosphates on them. I get a 3-carbon sugar with no phosphates because I, again, synthesized another molecule of ATP using this phosphate. This final terminal molecule here is known as pyruvate. You might recall from class that our next step is going to be to go on and oxidize pyruvate. As a friendly reminder to you, the fact here is that we have 2 G3Ps. So that means I'm going to make 2 NADH. I'm going to make 2 ATP here and 2 ATP here. So I'm adding into my equation 2 NADH. And I'm going to add 4 ATP. Remember that I was minus 2 here. So in the process of glycolysis, I have a net gain of 2 ATP per glucose. You will never be asked to draw this process from scratch. So let me remove just a little bit of my doodles here, leaving sort of the process behind, and show you what you might actually see on an exam to help you prepare for it. So let's get rid of some of our sugars and doodles. Okay. Okay, so if you were on an exam, the types of questions that you would see would be that you might come in and I would block off these 2 steps right here, and you're going to see a box appear in just a second in green. And I would ask you what goes into square 1 and what goes into square 2. And what you would need to do is pay attention to what comes before. So you know you're starting with a 6-carbon molecule, and what happens after. This is a 6-carbon molecule with a phosphate. This is the energy investment phase. So that tells me that I'm going to need to spend something in order to get my product that I want. And what you would need to do in order to get full credit on this answer would be to recognize that this is a molecule of ATP being hydrolyzed into a molecule of ADP. If I were to do the same kind of thing on the bottom, you might see, again, blanks that appear in this orientation. Once again, you would be paying attention to what comes before and after. Here I have a 3-carbon molecule with 1 phosphate, and it's being turned into a 3-carbon molecule with no phosphate. So something here needs to be gaining a phosphate, and in the energy payoff phase, in this particular step, that means that this molecule of ADP has been turned into ATP. I might also ask you a question about this step. For example, could you name the process by which this molecule of ATP is created? Since it's catalyzed by an enzyme, you could tell me that this is substrate level phosphorylation. I might also ask you to tell me about the molecules that are catalyzing this reaction. You might see a box appear for one of our two steps for which you know an enzyme, like this, asking you for the enzyme that catalyzes this reaction. And here I would want you to be able to tell me that this is a triose phosphate dehydrogenase. Or I might not give you this as your blank. I might give you that there's a triose phosphate dehydrogenase that catalyzes this reaction and ask you to tell me instead what would go up here at the arrows. Now, in your previous arrows, every time you saw a molecule before that was a 3-carbon 1-phosphate molecule that turned into a 3-carbon 2-phosphate molecule, you can say, oh, I needed to hydrolyze an ATP, but I'm in the energy payoff phase. The enzyme that catalyzes this reaction is a dehydrogenase, and the source of that phosphate is being provided here. It's an inorganic phosphate. So the phosphate isn't coming from ATP, which means that this step is going to be the one where it's NAD plus is being reduced into a molecule of NADH. By the way, that's another source you might ask you whether or not this molecule is being reduced. So if NAD plus is being reduced into NADH, you could also tell me that another molecule is being oxidized. There's a lot of different pieces here to know, but the trick to being able to handle this information is to recognize that you're never going to be asked to provide the next step without the clues in front of you. What I want you to be able to do is understand metabolism so that if you see what comes next and if you see what came prior, you can figure out what would come in the middle. So let's do one last example. You might also be asked to do this with molecules. Now, you would be asked to do this looking at full molecules, like the page of your textbook. But when you provide me your answer, you will tell me how many carbons were in this molecule and how many phosphates were in this molecule. So if we were to take a look at this molecule, what we would see is that the molecule before it is a 6-carbon molecule with 1 phosphate. The molecule after it is a 6-carbon molecule with 2 phosphates. So we need to decide whether or not the step before it is when the second phosphate is added, or whether the step after it is when the second phosphate was added. Since before and after both have 6 carbons, we can feel pretty good that the number of carbons here didn't change. But what you need to pay attention to is that the step after it is going to have a molecule of ATP hydrolyzed to ADP. So this is the step where a phosphate is added. So that means that this molecule that I have in front of me only has a single phosphate. It's one of the rearrangement reactions where there's no addition or change to the number of carbons or phosphates. You will always get blanks, but I will never put so many blanks together in the same region that you can't, let's say, figure all of this out. And I won't put so many blanks that once you've put it all together, you won't be able to figure out how many molecules that were made. So you do need to know that for each molecule of glucose, we make a net of 2 ATP. Let me write this clear. So we make a net of 2 ATP. Because remember that we have minus 2 ATP here, and we have the addition of this ATP and this ATP, but each one is times 2. So we've made 4, spent 2, that's a net of 2 ATP. We also made 2 NADH and 2 of this guy down here, which is pyruvate. So this is where all of our carbons are. We started with 6 carbons and we ended with 6 carbons because we have 3 times 2. All right, guys, keep studying, keep drawing, keep testing yourself this way. So cut out some pieces of paper, cover over one of the steps, any one of the steps on this page of your textbook, and see if you can fill out the information that would have been at that step. This is an example of where having a study buddy is super helpful. If you're the one who's covering over that step, it can be tempting to look before you do. Instead, have your study buddy place a couple of Post-it notes over top your textbook, or online, go in and actually create blanks for each other, share the documents, and test each other to see how many of them that you're getting right. And don't forget, if you like doing this kind of stuff, we can keep doing it. You can come to office hours, send me an email, keep watching videos, and keep at it. You guys are doing great.